Hello and welcome to my short talk. It's a great pleasure for me to be here and to be able to present the work in my group to you. I came to Dundee in 2013 and my group specializes in computer simulations of membrane proteins. And I'll briefly introduce my main interests to you, which are ion channels, in the following talk. The human body consists of trillions of individual cells. Each of our cells is bounded by a cell membrane, a thin layer of lipid molecules or fat-like molecules, and proteins immersed in this layer. Any communication that happens between the cells from one cell to another must traverse this membrane layer. A closer view of the cell membrane shows you some proteins that perform important functions in the membrane. Especially ion channels and transporters create the unique electrical properties of our cells and tissues. Each cell is defined by solutions of ions, that is charged particles, dissolved in water, and these solutions differ substantially between the outside and the inside of the cell. Ion channels and transporter proteins serve to create and uphold these ion gradients that are crucial for life. The function of ion channels is to allow electric currents, in this case ion currents, to flow between the outside and the inside of the cell. Each individual protein on the nanoscale contributes a current of about 10 to the minus 12 ampere. The combined actions of all the ion channels in a cell turn every living cell into an electrically charged compartment by creating the membrane voltage, which lies on the order of minus 50 to minus 100 millivolt. The membrane voltage is a defining feature of living cells, and it's required to keep many of the processes of life working. All cells in the living world are electrically charged in this way, but electrically excitable cells are a special form of cell that can change their voltage rapidly. This rapid change is used to communicate from one cell to another in our body. Electrical excitation, for example, underpins the transmission of signals in our brains and in our nervous system. Electrical signals also control the motions of our muscles. The rapid change in membrane voltage from negative to positive values and back in electrically excitable cells is called the action potential. It travels like a wave from one end of the cell to the other, spreading the electrical signals across our bodies. Ion channels are responsible for our senses. The example shown here is the sense of touch. Current flowing through ion channels is crucial to start and maintain the electrical signals in cells. The channel on the right is the channel that underlies our sense of touch. It converts force or mechanical deformation of our cell membrane into currents that are transmitted to our brain and felt as touch. Another example where ion channels are crucial is our sense of hearing. Sound waves generate a deflection of hair-like structures in the hair cells of our ears. These deflections, again, open ion channels that electrically excite auditory neurons. Ion channels are also crucial to enable our hearts to beat. You will all have seen electrocardiograms or ECGs, and those reflect the action potential of our cardiac cells. Here, the orchestrated opening and closing of sodium, calcium, and potassium channels enable our cells in the heart to beat at the same time and thereby keeping us alive. In my group, we study how these important proteins work. We study the mechanisms of ion channels that underpin the function of the brain, nerve, and our sensory system through computer simulations. We have developed methods to elucidate the channel function in real time under membrane voltage, atom by atom. This video shows you an example of a potassium channel. Here you see ions being conducted through the channel and inducing a current across our cell membrane in real time, and you see individual ions going through the pore. Each of the purple spheres is one individual ion leading to a microscopic current in the membrane of a cell. The computer simulations allow us to follow what happens under realistic timescales and realistic conditions like voltage across the membrane and study the mechanisms that underlie channel function. They also allow us to design drugs that intervene with channel function or change it in order to therapeutically approach the drug targets of ion channels. An example of my recent work is in the field of potassium channels, which are ultra efficient, but at the same time strictly selective for potassium. To enable the fast signal transmission in our nerves and muscles, action potentials must be very sharp and short. This requires maximum conduction speeds of the channels and a strict separation between the flow of different types of ions. Our recent simulations have solved a decades-long puzzle on how nature achieves this combination. As I mentioned before, computational simulations and computational drug design is also important to develop drugs against important channel targets. This is an example of an ion channel that sits in coronavirus. We are presently conducting simulations and computational drug design to develop inhibitors of ion flux in the coronavirus ion channel in the computer. This viral ion channel is essential for the virus to replicate. Similar drugs were shown to inhibit replication of influenza virus. In fact, the first known antiviral drug targeted the ion channel of influenza. In coronavirus, this channel is known to show very little mutation in contrast to other viral components, which makes it a very suitable drug target. In summary, ion channels are fundamental players in human physiology and in the function of all living cells. They serve as important drug targets, including for antiarrhythmics and antivirals. The team in my group has developed simulation techniques revealing how ion channels work at their most fundamental level, that is, at the atomistic level, atom by atom. This insight is now used to design drugs in the computer that maximize the impact of the drug candidates on target function. Thank you very much for your attention to my talk.